Good morning, everyone, and thanks for joining our webinar session. I might ask you to wait one more minute to make sure all the attendees have been able to enter the webinar session. So thanks for your patience. We're gonna start. So good morning again. Thanks for joining our webinar session. Let me introduce myself first. I'm Anada Rocha, European Life Science Commercialization Manager at Aventor, and I'm very pleased to welcome you to today's webinar, Small Volumes, High Requirements, Liquid Handling in PCR, given by Brandt. Today's speaker will be Dr. Mariella Neuhäuser. Mariella has a Ph.D. in human genetics and has been working at Brandt since 2011. But before I hand over to our speaker, I would like to give you some information about the progress of this webinar. It will be recorded and you will receive the replay link by email following this session. There will be a Q&A session following the presentation, but it will not be a live one. You should submit your questions by writing them down in the question box in the control panel at the right-hand corner. You can write them down at any time, even during the presentation, and they will be read and answered during the Q&A session. One last point, there will be a survey at the end of this session, and may I ask you to fill it in? This is very important for us to get your feedback in order to improve the quality of our webinars. And finally, if you have additional comments, requirements, you can always contact us at the address you're seeing right now on the screen, webinar@aventorsciences.com. This said, I hand over to you, Mariella.
Thank you very much, Anna, for the very warm welcome. Good morning, everyone, and thank you for joining today's webinar with the topic Small Volumes, High Requirements, Liquid Handling in PCR. As Anna already said, please ask questions in the question box, and the incoming questions will be answered at the end of the presentation. Let's just run quickly through the agenda. First of all, I'll start by looking at the PCR basics and methods. Then I want to illuminate liquid handling challenges in PCR sample preparation. Then we go on with the important question, how to choose the right liquid handling device. Another important aspect are the requirements to PCR consumables. We also want to find out when it makes sense to think about automation. And finally, let's have a short summary of the most important aspects. What is PCR and what are possible applications? PCR has become one of the most valuable techniques currently used in bioscience, diagnostics, or forensic science. This in vitro technique targets and amplifies specific regions of a DNA strand and makes millions of copies of the target piece of DNA. PCR and its variations have a wide range of specialized applications. One field of application is the environmental biology. PCR allows specific target species to be identified and quantified, even when very low numbers exist. So, for example, the detection of pathogens as coliforms in drinking water. It also plays a very important role in medicine. Some viruses, bacteria, parasites, and fungi which cause infectious diseases can be identified using PCR. Also in molecular diagnostics and biochemical analysis, PCR is crucial. as only tiny samples, for example hair, blood, urine, or other substances secreted by the body are necessary to identify DNA profiles, PCR is an important tool in forensic science. In genetic research, PCR is also used as an essential tool as well as in phylogenetic studies, where ancient DNA from sources as hair, bones, and other tissues can be amplified. In the field of food and agriculture, genetic manipulation can be targeted, and genetic technologies include a range of techniques that enable the modification of existing organisms for the purpose of improving foods and food production. Let's quickly have a closer look at the process of a PCR. The amplification of DNA via polymerase chain reaction technique was invented by the American biochemist Kary Mullis in 1983. Together with Michael Smith, he was awarded the Nobel Prize in Chemistry in 1993. The PCR is an enzyme-based process for duplicating DNA in vitro. It comprises 3 steps: denaturation, annealing, and extension. During denaturation, the reaction mix is heated over 90 degrees, and the double-stranded DNA is divided into 2 single strands by disrupting the hydrogen bonds between complementary bases. In the next step, the annealing, the primers anneal at the single strand between 50 and 60 degrees. The optimal annealing temperature depends on the length and the sequence of the primers. Longer primers and a higher number of guanine and cytosine pairings with 3 hydrogen bonds require higher temperatures than shorter primers and a higher number of adenine and thymine pairings with only 2 hydrogen bonds. The temperature is very important, as it has a big impact on the efficiency and specificity of the PCR reaction. During the last step, the elongation, the PCR mixture is incubated at the extension temperature from 70 to 72 degrees, which is ideal for polymerases. During this step, the primers are extended by incorporation of free dNTPs by the DNA polymerase, for example, Taq polymerase. Different PCR methods can be distinguished. Standard PCR or endpoint PCR allows a qualitative to semi-quantitative analysis after the sample is applied on the gel of the gel electrophoresis. Thanks to the uniform sample preparation, standard PCR technique is simple to understand and to use. Furthermore, it is a quite cost-effective technique. Amplification of long DNA sequences up to 3,000 base pairs is possible. With help of certain polymerase mixtures, further additives in the PCR reaction, and optimal conditions, even fragments longer than 20 to 40 kilobase pairs can be amplified. However, the efficiency of amplification decreases as the target DNA gets longer. To amplify RNA, a so-called reverse transcription PCR can be performed. In the first step, the reverse transcriptase converts the RNA into a complementary DNA. This so-called cDNA is then used as a template for exponential amplification using PCR. Quantitative PCR, short called qPCR, allows real-time detection of DNA amplification with high sensitivity. This method allows reliable detection and measurement of products generated during each cycle of PCR process. Standard PCR is often used for downstream applications like for genotyping, cloning, or preparation of DNA libraries. qPCR has a wide range of application, for example for microRNA analysis, SNP genotyping, gene expression, analysis of forensics. Summing up, the endpoint PCR technique is a qualitative method to amplify the gene, while the qPCR technique is a quantitative method to amplify as well as to measure the target gene. Of course, there are many more different methods next to classic PCR. For example, hot start PCR where the polymerase is blocked by thermolabel antibodies. The PCR starts with approximately 5 minutes at 90 degrees to activate the polymerase. Hot start PCR is used to optimize the yield of the desired amplified product in PCR and simultaneously to suppress non-specific amplifications. Another variant is the nested PCR, which includes consecutive PCR reactions of 25 cycles. The first PCR uses primers external to the sequence of interest. The second PCR uses the product of the first PCR in conjunction with one or more nested primers to amplify the sequence within the region flanked by the first set of primers. The big advantage is the very low possibility of non-specific amplifications. Multiplex PCR allows simultaneous amplification of many targets in one reaction by using more than one pair of primers. This saves time, but there is a high risk of cross-template binding. Droplet digital PCR is a method for the absolute quantification of rare alleles. Samples were segmented by using water and oil emulsions to create droplets from which their genetic material can be identified and quantified. The sample is splitted into droplets in which the individual PCR reactions can take place. And of course, there are many more different methods. Each partition contains only one target molecule or no target molecule, as well as a specific fluorescently labeled probe for each target. The reaction mixture is then read out. That means the fluorescent droplet with PCR product and the non-fluorescent droplets without PCR droplet can be counted to determine the concentration with a reading device similar to a flow cytometer. Droplet digital PCR is a further development of quantitative PCR. The advantage is that a higher specificity and sensitivity can be achieved compared to conventional quantitative PCR. Now, here is an example which illustrates three main requirements for PCR. These are reliable results, small volumina, and short cycle times. The coronavirus is still, or better said again, on everyone's lips. And here too, PCR plays a crucial role because it enables reliable detection of the virus. As an example, here is an excerpt from the test kit of the company Microgen Diagnostic with a total sample volume of only 25 microliter. This kit works with a real-time multiplex reverse transcription PCR technique. Reliable PCR results are essential to monitor the spread of the virus as closely as possible, and false negative results must be excluded under all circumstances. Therefore, the test must be reliable and highly sensitive to provide a correct result, even with small sample volumes. In addition, small volumes are essential to be able to work economically and also with little sample material. And as a third point, I would like to emphasize the cycle time because, as we all know, time is money. And in order to be able to examine as many samples as possible quickly, a short cycle time is just as indispensable. To achieve this, among other things, the thin wall and associated thermal conductivity of the PCR tubes is important. This example illustrates the demands placed on PCR today and what needs to be taken into account. The basis for meeting these requirements is high product purity, fast and safe processing, and a secure closure to prevent contamination and evaporation. And it is precisely these 3 points that are often underestimated, which is why I would now like to go into more detail. When preparing a PCR, it is very useful to create a master mix. The master mix includes a mixture of PCR reagents at optimal concentrations that can be prepared and divided among many PCR tubes or PCR plates. A master mix speeds up your pipetting. You save a lot of time and reduce pipetting errors. as all preparations contain the same mix. This improves the reproducibility of the experiments. The Mastermix includes the thermostable DNA polymerase, most common, the Taq polymerase, a mix of the 4 different D-antipees, and a cofactor factor for the DNA polymerase, usually magnesium chloride. Furthermore, it contains a buffer which proves a suitable chemical environment for the DNA polymerase. A Mastermix minimizes sample-to-sample and well-to-well variation and improves reproducibility. It is important to prepare a bit more than the required volume. This ensures to have enough volume, even in the case of pipetting inaccuracies. Sample loss may occur because enzyme solutions like the Mastermix contain detergents. Detergents are likely to foam, which might lead to inaccuracies during pipetting. On the next slides, we take a closer look on how we achieve more accurate results when pipetting buffers or viscous liquids like the Taq polymerase. But regarding our Mastermix, finally, we have to add template DNA, which contains the target region and primers. The PCR preparation should be done on ice, for example, with help of a cooling block. For qPCR, dyes like cybergreen and strand-specific probes must be added. Always add a positive control and a no template control with your PCR run to ensure the reliability of your results. The challenge for liquid handling even starts in the extraction of the DNA or RNA from the sample. The next step is the transfer of a very small amount of extracted DNA or RNA with high accuracy and precision for reliable results. The success of PCR depends on the precise amount of reagents that are added to the reaction mixture. A slight variation in the amount of any of the reagents can lead to significant changes in the final results. For example, if the amount of DNA template is too low, the PCR may fail to amplify the target sequence, whereas too much template can lead to non-specific amplification. Similarly, the addition of too little or too much enzymes, primers, or nucleotides can affect the efficiency and specificity of the reaction. Therefore, it is essential to ensure that the reagents are accurately and precisely dispensed. There are different devices which might help to prevent mix-up samples and which make work more efficient, as for example multi-channel pipettes. Liquids from plates are transferred like in the picture on the right hand. A further challenge is the mix of liquids with different physical properties. On the one hand, aqueous solutions are pipetted, but also Taq polymerase, which is more viscous, has to be added without pipetting errors. Intense, repeated operations done on mechanical instruments without proper ergonomic design result in prolonged stress that can lead to a number of muscular problems, known collectively as repetitive strain injuries. At particular risk are the muscles in the neck area, shoulders, arms, and thumbs. Thus, laboratory work is often accompanied by the appearance of, among others, tendonitis and carpal tunnel syndrome. Especially with microliter pipettes, the need for fatigue-free operation is crucial. Therefore, use pipettes with low operating forces and a short stroke. There are several methods for liquid handling in PCR, including manual pipetting, electronic pipetting, and liquid handling robots. Which pipette should be used for the preparation of the PCR? When does it make sense to use a manual single-channel pipette? Single-channel pipettes suit best to aqueous liquids in the microliter to milliliter range for small to medium sample sizes. But also when adding, for example, individual template in wells of PCR plates, a single-channel pipette is the best choice, as it shows high precision and functionality and is therefore a standard device in routine labs and in research. The tips are used only once. This is particularly important for applications where sterile conditions are required or no carryover is allowed. Multi-channel pipettes work like 8 or 12 single-channel pipettes in a row. Therefore, 8 to 12 pipetting operations can be carried out simultaneously. 96 well plates with standardized spacing can be filled very efficient. Multi-channel pipettes are also ideal for serial dilutions and for washing of microtiter plates. Summing up, manual pipetting is a common method used in many labs. However, it can be time-consuming and is prone to human error. To minimize errors, it is important to use calibrated pipettes and compatible tips and to carefully follow the protocols for dispensing the reagents and pipette as consistent as possible. Pay attention also to the angle, the depth of immersions, and so on, because this also has an impact on the results. Electronic pipettes can improve the accuracy and precision of liquid handling because they dispense precise volumes of reagents and are less prone to human errors. The combination of motor-controlled pipettes and ergonomic design enables stress-free, fatigue-free operation. It also reduces the demands on the thumb while carrying out lengthy series, which would otherwise increase the risk of the repetitive strain injury syndrome. An additional advantage is the execution of pipetting programs, such as the gel electrophoresis mode, which precise display of delivered volume, reverse pipetting, mixing, and a dispensing mode, which are not possible with manual pipettes. To dispense the reagents in a specific order and volume with help of the different programs, the risk of errors can be reduced. Another advantage is the high reproducibility, as operation forces do not have an impact on the volume. How important is the nominal volume of the pipette? Can I use a 10 microliter pipette for dispensing 1 microliter of DNA? In general, adjustable volume pipettes offer greater flexibility than fixed volume pipettes and can be used for many different applications. However, their accuracy is highest at the maximum pipetting volume, and it decreases slightly with the pipetting volume. What does that mean in numbers? When we have a closer look at the pipette with a volume range 0.1 to 2.5 microliter, we can see an accuracy of 1.4%. That means plus or minus 0.035 microliter when pipetting the nominal volume. When half of the nominal volume are dispensed, the accuracy is reduced from 1.4 to 2.5%. When pipetting even less, for example, 0.25 microliter, the accuracy continues to decrease to 12%. And this accuracy completely fulfills the ISO requirements. This example shows that it really makes a difference to work in the upper or lower volume range. This should be taken into account in the choice of the pipette. What are the differences between air displacement and positive displacement pipettes? The air displacement principle is used mainly for pipettes and enables precise dispensing with fast and easy handling. By moving the piston in the pipette shaft up and down, a negative or positive pressure is created. This causes liquid to be aspirated or to expelled from the tip. However, the liquid is always separated from the inner piston and the pipette shaft by an air cushion and does not come into contact with the pipette itself when handled correctly. This enables contamination-free yet simple handling with the highest precision. But this is also where the application limits of the system lie. Media that strongly influence the air cushion can affect the accuracy of the result. For example, foaming liquids like detergents, we already told about it before, make precise work difficult. The positive displacement principle is often used for repetitive pipettes, or specialized positive displacement pipettes. In contrast to air displacement systems, the piston of the positive displacement pipettes is in direct contact with a liquid to be dispensed. The piston aspirates the liquid through a vacuum and wipes the walls of the tip clean during dispensing, down to the last drop, which clearly leaves the tip. This principle ensures highly reproducible results regardless of the liquid and the influence of an air cushion. The piston, which is built into the device in an air displacement model, is located in the tip cylinder in positive displacement models. This plays a role, for example, for the extraction of DNA from whole blood. In summary, air displacement pipettes are the right choice when tips must be changed all the time, as for example when adding different DNA to the different wells. Positive displacement pipettes are better for pipetting foaming or viscous media and preventing cross-contamination, as aerosol contamination can also be an issue not only with pipette tips but also when using filter tips. In the last slides, we discussed the different liquid handling devices. Let's now focus on other important aspects, the PCR consumables. The quality of PCR consumables is very important. What is the worst case if purity and cleanliness of the workspace is not guaranteed? In the lab, purity is not the same as cleanliness. Even tiny particles as unwanted additives can lead to false positive or false negative results. Also, modern cleaning agents are available for cleaning the workplace, also in the case of consumables. Take care to avoid contamination also in the production stage and ensure that appropriate raw material has been used. The top priority is therefore to ensure that the work area and the PCR products used are free of DNA, DNAs, and RNAs, so that the samples to be tested are not contaminated or destroyed. These material criteria are met at Brand and tested and confirmed by independent labs. Not every factor that can influence your experiment is obviously detected. For example, if you measure the DNA content of a sample and the spectrometer outputs a positive value, you probably assume that the measurement was successful and is correct. Already in 2008, scientists noticed that the use of different plastic consumables had an influence on the experimental results, and that this was due to substances contained in plastic consumables. This observation was published again several times in the following years for various applications. The different manufacturers provide various guides to the classification of purity, which help to make the right choice of products. Don't worry, this scheme should only give you a rough overview of the different purity levels. In addition to the general certified quality label, there are special quality levels for PCR, cell culture, and liquid handling. As a result, you will quickly and easily find the right products for your application. The right quality level of PCR consumables is required for reliable and correct results. When source materials are selected for life science products, Brand ensures that any substances which could leach out of the raw material and influence the biological tests are reduced to the minimum necessary for processing. Mold release agents are not used in the production process. For the production of pipette tips and PCR products, Brand uses specially selected polypropylene types which are free of the additives dihemda and oleamide. These two additives, which are often found in polypropylene granulates, can interfere with biological tests and lead to false results. Manufacturers like Brand offer cleanroom quality PCR consumables for sophisticated PCR and qPCR applications, where quality and purity are key. On this slide, you see a picture of our cleanroom. Live monitoring of our validated ISO Class 8 cleanroom and a high degree of automation ensure the purity of PCR products And independent test laboratories accredited PCR products for the absence of DNA, DNAs, RNAs, and endotoxins, as well as pyrogens. Next to the quality level, fast temperature transfer is crucial for PCR plastics. The ultra-thin-walled PCR plate design facilitates constant, rapid, and precise heat transfer, leading to convincing yields and a short PCR cycle time. In addition, the smooth vessel interior minimizes the binding of enzymes and nucleic acid to the walls. For automation, plates with a rigid frame for high stability and thin-walled wells are a good choice. As the reaction volume is very small, it is necessary to avoid evaporation. This picture shows the percentage evaporation losses of different PCR systems. In this case, we used a 96 PCR plate in combination with an adhesive sealing film. As you can see, there are really differences in the PCR consumables. Therefore, it makes sense to optimize not only the PCR reaction mix, but also the choice of PCR plastics. In the picture on the right side, the remaining volume after a 4-hour PCR program is shown graphically. Especially in the edge regions of the plates, in the red marked fields, the evaporation loss is up to 33%. This study also illustrates how important it is that the combination of the PCR plate and sealing film are a compatible system. And moreover, an optimal lid pressure of the thermocycler is required. Furthermore, when using PCR tubes, please inform how many tubes must be used at minimum, because too less tubes might be damaged by the lid of the thermocycler as the pressure might be too strong. Another important point is the fast identification and avoidance of confusion of samples. Colored vessels help to differentiate with project-related colors and allow easy visual sample tracking. We also provide PCR strips with detached cap strips, which can be opened contamination-free with grip tabs on the end and side of each cap. The wells are numbered for clear identification. On PCR plates, alphanumeric coding and the cut corner help to identify the wells and minimizes the risk of confusion during pipetting. Elevated wells of PCR plates and tight closing caps or sealing films help to avoid cross-contamination. The secure seal prevents contamination to ensure reliable results, while also reducing costs due to minimized evaporation. Choose domed caps for extra sealing pressure via the lid of the thermocycler, or flat caps required for qPCR. Attached cap strips can be easily opened and closed with one hand, while separate cap and film strips are especially suitable for automation as they will not obstruct robotics. Considering the handling, holding the strap at one end and open it carefully. The cover strips on each cap have a small lip on the side for careful, contamination-free opening. The ease of the handling protects from spillover. Selecting the right sealing film for your application improves your workflow efficiency and the quality of the results. For PCR, the innovative self-adhesive press-to-seal sealing film wins over with easy handling. The encapsulated, pressure-sensitive adhesive keeps the film easy to handle and non-tacky to the touch. After sealing, areas above the sample wells remain adhesive-free and do not distort PCR samples. On top, the ultra-thin liner and high transparency allows detection of smaller signals during real-time PCR. To make it easier to find the right sealing film for your application, please use selection charts, which are often provided from the manufacturers. On this slide, this is an example from the sealing films from Brandt. For quantitative PCR, white PCR disposable for optimum results are available. With titanium dioxide white-colored products, reach an optimal reflection of fluorescence signals and prevent signal transfer throughout the thermocycler block. White PCR products yield significantly better results in qPCR than transparent vessels. In combination with a smooth surface, the white color ensures optimal reflection of the fluorescence signals. In combination, ultra-transparent sealing or flat caps suitable for qPCR should be used. On this picture, you see the very high fluorescence signal of white wells in blue compared to a significantly lower fluorescence level of transparent wells, here shown in green. Whether individual PCR vessels used for test validation or high-throughput testing of standardized processes with 96 or 384 well plates, the selection of the right consumable saves on costs and time and also minimizes waste. Therefore, before the experiment starts, plan which sample volume is necessary and which format makes sense in the experiment. PCR strips with a connector offer good flexibility. The strips can be cut easily using scissors or twisting them for individual adjustment. For applications using strips with increased rigidity, we recommend using tubes with 3 braces. These cannot be easily divided, but offer maximum security thanks to improved stability. Side grip tabs and attachments on the individual caps allow for easy opening and contamination-free handling. Strips with attached individual lids offer improved protection against contamination and reduce the danger of mix-up. If you are not sure which consumables and which setup suits to your experiment, this video gives you a short overview of available formats.
How to select a PCR plate. Selecting the correct PCR plate for your thermocycler can be daunting. To start your PCR plate selection process, you must know which thermocycler and block you'll be using. You must also ask the following key questions when doing traditional or real-time PCR. How many samples will you be amplifying? PCR plates are available as 24, 48, 96, and 384 well models. What volume samples will you be amplifying? PCR plates are available in standard profile, 0.2 milliliter, or low profile, fast models in approximately 0.15 milliliter volumes. What style plate would you like? Non-skirt for universal thermocycler fit, semi-skirt for more rigidity and easy grip, full skirt for the ultimate in PCR plate sturdiness and is compatible with automation. What attributes does the block in the thermocycler have? Each thermocycler has a block with different shapes, well counts, and potentially different cut corners that should match up for the best fit. Will your application be traditional PCR or quantitative real-time PCR? Clear PCR plates can be used for traditional or real-time PCR, but white PCR plates are optimized for signal detection during quantitative real-time PCR. Some manufacturers also have color alphanumeric embossing that allow for easy sample tracking. Lastly, always consider your sealing methods. Small volume samples can evaporate during the repeated heating and cooling process of PCR. To prevent precious sample loss, select PCR strip caps, sealing mats, or sealing films.
Basically, we can make a distinction between the optimization of the methodical approach and the optimization of the working material used, whereby both points are absolutely relevant for an optimal result. The optimization of the methodological approach often requires profound scientific background knowledge and includes aspects such as the selection of a suitable polymerase, the correct primer design, the adjustment of substrate and product concentrations, the avoidance of interfering substances, the control of environmental conditions, and, of course, the optimal PCR program. But these factors must be taken into account in advance when establishing the method. So let's sum up. What is important when choosing the right liquid handling device? As in PCR, the volumes are usually very small. It is important to pay attention to the precision of the pipettes. In case of high sample volumes, also have a look at the ergonomics to avoid long-term injuries and think about automation if possible. Use repetitive pipettes to difficult liquids like DMSO. To avoid evaporation loss, sealings or plates should be compatible. By selecting the right materials and observing a few criteria, you can quickly achieve better results. With help of easy removable seals and easy to open lids, spillover can be prevented. For upscaling, different formats are possible, like tubes, strips, or plates. In order to prevent a premature reaction, the PCR reaction mix should be prepared on ice. And as already mentioned before, we recommend white PCR consumables for better signal detection. The next point I want to address is the question why subsequent sterilization of PCR consumables is not advisable. We do not recommend it because the radiation sterilization usually used damages the very thin-walled products and increases evaporation loss, in addition to the embrittlement of the products. Autoclaving is not recommended because autoclaves are a source of contamination for these high purity materials. During autoclaving, fragmented DNA molecules from microbial debris can be released by the steam and easily contaminate materials and surfaces. As briefly mentioned before, there are special PCR plates for automation. The plates combine the best of two materials. The skirt is made of polycarbonate for exceptional stability and smoothness. This is ideal for automated use with robots, as the plates do not deform. Heat sealing is no problem, as the plates do not shrink or wrap, and evaporation loss is minimized. Therefore, lower vacuum volume is also possible. The wells of these plates are made of medical polypropylene. Due to the rigid frame, the wells can be formed extra thin with uniform wall thicknesses. This ensures short cycle times and reduces the risk of cross-contamination. To give you an impression of the stability, I would like to show you a short video where we tested the plates on our own. Before you decide to automate your PCR, there are a lot of aspects which must be considered upfront. A first point is the complexity of the software. There are many automated liquid handling devices which address different requirements on the market. Therefore, it is important to define your needs before you start to choose an adequate system. The procedure needs to be defined with high precision in advance. Make clear which steps should be automated, which labware is necessary, are there any possibilities to change some steps of my manual procedure in a way that the automation can be done? Do I have to work under sterile conditions? How stable are my liquids? How do I handle the residual liquids, especially expensive liquids in the labware? What tolerances are needed, and so on. And of course, you have to predefine aspiration and dispensing speeds in relation to the liquid type. There is often the question, when does it make sense to think about automation? First of all, let's have a closer look at the main differences between electronic pipettes and automated liquid handling systems. Of course, you have to program the method with the automated liquid handling system. That means the setup time can be longer the first time. We recommend to test the setup before using the PCR samples. Nevertheless, there are many advantages of an automated system. The method is always identical, and there is no personal influence. The personal influence has an enormous impact on the results. The process is documented. That means the method can be saved, modified, and sent easily. The liquid handling station is widely used for low to medium sample throughput, and it closes the gap between manual pipetting tasks and big, fully automated workstations. It provides maximum flexibility for small and medium sample throughput and varied pipetting tasks. Method creation is very simple and can be done within minutes. Thanks to the intuitive software, no programming knowledge is needed. The liquid handling station pipetting robot handles all your pipetting routines, leaving you more time for other work. Automation is worth it, even for small and mid-sized sample volumes. Thanks to its compact design and the front door, the liquid handling station needs very little space. It works with single vessels, PCR strips, reservoirs, 96 well and 384 well plates, as well as the brunt insert system. It is an open system. That means that also disposable items like microplates, single vessels and so on from other manufacturers can be used, but it is necessary to use brunt tips. And as already mentioned before, the intuitive software needs no programming skills. This ensures simple method creation within minutes. PCR with this automated liquid handling system is quite simple. The workplaces can be freely assigned, including PCR tubes and strips, microcentrifuge tubes, reservoirs, and plates. This makes it easy to create new methods when other applications should be established. A PCR application, for example, can be managed with only 2 transfer steps from 1 to 96 samples. All of the customer's labware can be used at the workbench, including sample racks and cooling blocks for PCR sample plates. Summing up, we can say that medium-sized throughput sample preparation can be easily automated with this system. And with a modular design, it is open for new applications in the future. Let's just briefly recapitulate the main arguments for automated liquid handling systems. Especially with monotonous and error-prone work steps like pipetting and diluting, a robot really demonstrates its strength. Once defined, a method can be reproduced over and over again with exact precision. With help of the liquid handling station, for example, you save up to 70% of time compared with manual pipettes, and you can do other work during the PCR preparation. With an automated system, you also achieve better results for accuracy and coefficient of variation. Let's now summarize the outcome. Regardless of whether you work with an automated system or manual electronic pipettes, accurate and precise liquid handling is essential for reproducible results in PCR. Manual pipetting is a common method used in many labs, but it can be time-consuming and is prone to human errors. Electronic pipetting and liquid handling robots are alternative methods that can improve accuracy and precision of liquid handling. Regardless of the method used, it is important to follow the protocols carefully and to use calibrated equipment to ensure accurate and precise dispensing of reagents. Make sure that your PCR consumables are free of substances which interfere with your PCR products. Reduce the contamination and evaporation risk for reproducible results. So now I'm coming to the end of my presentation. Thank you very much for listening. And now we will start the Q&A session. Please ask your questions in the question box. Thank you. So Benjamin, are there any questions available already?
Yes, there is a question from Sarah. Hello, Mariella. For my pipettes, I use tips from other manufacturers. Does this have an impact on the accuracy?
This is really a very good question because, yes, it is most common in the labs that you do not use the original tips which are compatible with your pipette. And the pipette and the pipette tips are a matching system. Therefore, if you send your pipettes to a calibration service, then please make sure that you also send your third-party tips with your pipette so that the service can check if the tip and the pipette match, if it is a matching system. Because if the tip cone and the tip do not fit perfectly, there is a leakage in the system. And if you have a leakage, then you do not dispense the right volume. Yes, and for this case, we also have a little benchtop device. It is called pipette leakage testing unit. And you can ask your VWR counterpart who gives you more information on that, or you leave your email address, then we can send you our information. And with this pipette leakage testing unit, you can see within seconds if the pipette and the tip fit together, or if there is any leakage, and then you have not the right accuracy. Yeah.
Thank you, Mariella. Another question from Sebastian. If I have critical samples which should be dispensed in the laminar flow hood, can I use a small liquid handling robot, or do I need another system?
So if you have samples which have to be protected from particles and microorganisms, we also recommend the liquid handling station, but it is called liquid handling station flow. And it has a flow box which filters the air and directs the filtered air in laminar horizontal layers across the plates. And yes, and the volume will the air will be filtered, and the air will be replaced 260 times per hour inside of the device. But we would like to send you more information about that, and also talk about your needs and your application to give you really the best advice. Please just leave us your email address or your phone number, and then we will contact you.
And so a last question. In my PCR, I add 1 microliter of DNA. Which pipette do you recommend?
Of course, as we already saw in the webinar, the best accuracy is achieved with a 1 microliter pipette, but also a 2.5 microliter pipette is totally okay. So you are within the limits of the ISO tolerance. So that would be great. Yeah.
No more questions in the chat. Thank you, Mariella.
Very, very welcome. And then I would like to give over to Anna again. Thank you.
Thank you, Mariella. Thank you, Benjamin. Thank you for your very comprehensive presentation, very clear, and thanks for answering the questions. To all our attendees, as said during the introduction, this webinar has been recorded. You will get the link by email together with additional documentation, such as application notes and product information related to this webinar. But you can also access the replay of the webinar via Aventor's website in our webinar corner, where it will be posted and remain available for 1 year. So you can go through at your own convenience, even share it with people that might be interested and that were not present during this session. I hope you find this presentation interesting, helpful, that it has met your expectations. Please do not forget to answer the short survey before leaving, and thank you very much. Wish you a very nice day. Thank you again, Mariella and the brand team, for a great presentation.
Thanks a lot.